All right. So, welcome to the last Zoom talk in this year's series, uh, Fayette Prairie Chapter of NPAT series on the impact of climate change on our native prairies. The talk we had scheduled for December with Dr. Russell Fagan from the Department of Ecology and Conservation at Texas A&M is rescheduled for February 13th, 2025, and we'll put that in the chat this evening as well. So make a note on your calendar. We'll be kicking off 2025 with Dr. Fagan's talk about the future of restoration in a changing world. So stay tuned. Tonight, I am happy to introduce Dr. Elizabeth Bowman. Dr. Bowman is a fungal ecologist specializing in the relationship between plants and fungi. Her research has focused on the fungi that live in soils, mycorrhizal fungi, and the fungi that live in plants, endophytic fungi. And she has studied plant and soil communities in various ecosystems, including those isolated mountain ranges in Arizona called Sky Islands, as well as mesquite savannas in Texas and Kenya. Currently, she is leading some innovative research at Hiro Technologies in Austin, Texas. That's Hiro, H-I-R-O, where she is developing an approach to break down and degrade plastics using fungi, which is super interesting. That will be going public in January. And so she will be able to talk more about it and educate people about this whole process then. Until then, we just know that she's doing some very important research. She is passionate about citizen science, and I think a lot of mycologists are, and science communication. She does a lot of talks as well as mushroom walks with various groups in Austin, and her husband and two daughters. She is from Nacogdoches, Texas, and she has lived all over the world. She and her husband were both in the military, so they've been all over. So, Dr. Bowman, we really appreciate your research, and we appreciate you being willing to talk to us tonight about what's going on in our soil microbial communities.
Yeah, thank you for having me. I was really excited to do this talk. Let me share my screen. I didn't know I was going to be the last, the last person to give the talk this series, but I hope it's good. Way to end for you guys. Okay, does that look okay for everyone? Okay.
It does.
And I have this caption on the bottom that will capture everything I say. If that bothers anyone, I can turn it off. Okay, so today I'm going to be talking about the soil microbiome and processes that they are kind of take part in and how that might be changed by climate change. But before I really get into that, I kind of wanted to frame the exact communities we're going to be talking about. A lot of times people say microbial, but you're actually including two very distinct communities within that title. So Tree of Life starting very broad tonight. So we have our three domains of life, bacteria, eukarya, and archaea here. So when we talk about the microbiome, in general, we're talking about bacteria, which is its own domain, and then fungi, which are within the eukaryotes. And we'll talk about them a little bit more in depth. But bacteria are very abundant. Obviously, there's estimated to be up to 1 billion species. But only about 30,000 have been named and their function has really been described. So there's quite a bit of information we do not know about bacteria still. Oops. Okay, so zooming into eukarya, we have our three main kingdoms within eukarya: plants, fungi, and animals. And I like to emphasize this because I love fungi, but a lot of people are not aware that fungi are their own kingdom. And so when you think about how diverse plants are and animals are, you will see the same amount of diversity of both form and function within fungi. So very diverse. And again, there's millions of species estimated, but only a small portion have been described. So fungi and bacteria, which make up the microbiome predominantly, although there are some other groups that are sometimes included, there's a lot we don't know. And a lot of that comes down to just difficulties in culturing them. Many of them are unculturable. And so we really need to develop better instruments. We've only developed the ones we have now the past maybe 50 years. So we're still developing our knowledge base within these groups. A big issue with studying them is that they're cryptic. You can't see them. So this is a photo from South Texas where you have mesquites and grasses, very dry, sandy soil. You can see the plants. If there were animals, I'm sure there are out here that we aren't seeing, but they are there as well. But the fungi and bacteria, it's not really clear where they will be. So where are they? So first place is just in soil. There's very abundant. 1 gram of soil can have millions, if not billions, of cells of bacteria and fungi, and they just are free living within them. The second group, the one I'm particularly in love with, are plant-associated ones. So these are picked up from the bulk soil. The plant has like a filtering mechanism where it filters in ones that it wants into its body. So into the roots, the rhizosphere, and then up into the leaves as well. So first group, which I'm sure everyone here is familiar with, nitrogen-fixing bacteria. So they induce the host plant to make these little houses on their roots that they live inside, and they take atmospheric nitrogen and fix it in a form that plants can use. The second group are endophytic fungi, and these are, I guess, fairly new compared to the other groups in terms of length of time that they've been studied. And they've been found in all parts of every plant studied. So they're very abundant and you can have multiple species within a single leaf. So yeah, they aren't necessarily pathogens. They don't cause obvious symptoms of disease. They can modify the phenotype of the host though. So there's been quite a few studies into how they do that. And then the last group, which I'm sure everyone also is familiar with, are mycorrhizal fungi. So the two main groups are arbuscular and ectomycorrhizal. Arbuscular is mainly what you're going to find within grasslands. So these actually grow into cells that are intracellular versus ectomycorrhizal, which are intercellular. And actually, arbuscular mycorrhizal fungi are very unique in that they are their own group within fungi, whereas ectomycorrhizal fungi are kind of found throughout fungi. So this is one single evolutionary event led to arbuscular mycorrhizal fungi. And this is kind of what they look like. Over here, you have the arbuscules of the arbuscular mycorrhizal fungi. This will be in the cell, and that'll be the interface at which the fungus is exchanging carbon and nutrients with the host plant. And you can see more in this one as well as these small storage vesicles. Over here, you have ectomycorrhizal fungi, and you can see it is an extension of the roots. So you have the roots of the plant end about here, and the rest of this is all mycelium, or the body of the fungus. And it's able to access nutrients and water further out into the soil so that the plant's not having to put those resources into making root material. So they're really important. And the last group are pathogens. And pathogens, I feel like, get a really bad rep because we see the damage that they do. And in agricultural systems, they do cause a huge issue. But in natural systems, they play an important role that I wanted to highlight, which is that they increase diversity. So when you have an ecosystem, a community such as a grassland community, you have really high diversity. And part of that is kind of driven by pathogens, where you'll have both how related another plant is and how abundant another plant is will lead to how much pathogen load there is. So for instance, if we're looking at these leaves right here, this oak tree, and here we're seeing the pathogen spillover from all the other members, but we see larger spillover from more closely related individuals, so like D here versus B here. And then also ones that have higher relative abundance. So this A plant here has more of a pathogen spillover than B, even though they're equally related to C. and F here has much smaller because it's much further in relation and it is high in abundance, but not as much pathogen spillover. So pathogens do play a role and they aren't always bad. Okay, so now we know where they are. They're in the bulk soil as well as the rhizosphere and phyllosphere, root and leaf areas of the plant. But what are they doing? So they play an important role in organic matter decomposition. So here are two forms of a log that you've probably seen while out walking. And the way it looks can tell you a lot about what's going on. This is one of my favorite facts to share. On the left, if you were to touch this log, it would come away really flaky and soft. It wouldn't really feel like wood, more like sawdust. And this is being degraded by a white rot fungus. So this fungus specifically attacks the lignin, which is the most recalcitrant, hard to decompose, naturally occurring structure in nature. This one on the right, which if you were to feel it, would be very hard. It would break off in these small cubes, which is kind of fun to do. This is being broken down by what's called a brown rot fungus, and it's only breaking down the cellulose and hemicellulose. And so you see kind of like this niche partitioning within the decomposing log, different fungi. Okay. Another really important role that they do is soil formation. So you have these aggregates of soil in really healthy soil that if you were to dig a hole and pull up a clump, it would just be this whole clod you can put in your hand. And this is done by both fungi and bacteria, and that they exude polysaccharides and proteins and metabolites into the soil. In addition, the fungi, their body is just these long threads that create hyphae that kind of glue the material together. And this structure is really important for creating niche space for microbes. Excuse me, and you'll have a much healthier community. And it's also important for nutrient and water distribution throughout the soil. And you can kind of see what kind of structure your soil has with what's called a slake test. I think that's right, where you have a mesh that you put your clod of soil into and then immerse it in water. And as it draws water up, if it holds its form, it's really great structure, has great aggregation of the soil, versus if it falls into solution, then it doesn't have very good structure. And then lastly, if not most importantly, they contribute to the nutrient cycle. So here are two cycles, nitrogen cycle and carbon cycle, that microbes really play a large role in. So bacteria here will, these rhizobia on pea plants will fix atmospheric nitrogen, excuse me, into the soil, and then different bacterial groups within the soil can change its form, eventually making it go back into the atmosphere at the end of its cycle. And then carbon, plants, as they drop leaves, drop litter, they'll lose this carbon to the soil. It'll either remain in the soil as kind of a reservoir or get converted by microbes and back on up. And fungi and bacteria, but especially mycorrhizal fungi, I want to emphasize, play a huge role in sequestering carbon below ground. So about 13.1 gigatons of carbon dioxide annually is sequestered below ground by fungi from plants. So a really large number. Okay, so coming to the meat of this talk, how will climate change alter these services that microbes play within our ecosystems? So I've kind of here summarized all the points that have been discussed over the past few talks. So we're going to see changes in temperature and precipitation. Temperature is going to go up. Precipitation is going to become more sporadic with longer periods of drought in between and harsher rain events. More soil evaporation, changes in plant phenology and the composition of plant communities, and then changes in the frequency and intensity of disturbances. So I'll talk about each of these in the context of microbial ecology, starting first with the climate-related changes. Okay. So, of course, precipitation and temperature are really important in structuring microbial communities, especially precipitation. Um, but this interaction is complex, as we'll see. So I tried to frame this with a series of cartoons, which I hope is okay, where we can look at how the climate will change, how that changes plant productivity, and then how that will impact the microbial community. And so some of these changes are directly from the changing climate to the microbial community, but I would argue, and I know that there are microbial ecologists who may not agree, that it is the plant community changes that are going to have a stronger impact on the microbial community. So with increasing precipitation and temperature, you're going to see an increase in plant productivity. And this means more litter will be going into the soil. You'll have more root exudates coming from the plant into the soil. It'll have more carbon available for relationships with microbes. So, they pay for these services from the microbes. If you have more carbon, that's more money to pay for these services. Okay. So, hang on, I went back too far. So you're going to see an increase in both abundance and richness, but there is a limit to this. Once you hit a certain level of precipitation, when there's too much water, you're going to have more of a anoxic, so low oxygen environment, which is bad for microbes, as well as you're going to have soil nutrients start leaching out. So some precipitation will be good, but there will be a limit to that. And it really depends on what system you're looking at about what that limit will be. But when we have the opposite, which we're going to see more of, which is drought, a decrease in precipitation, decrease in plant productivity, which will mean you have a decrease in microbial abundance and richness, and really you'll have a selection for those microbes that are really drought tolerant. So similar to plants and animals, there are microbes, fungi, and bacteria that are more adapted to different environments. There are drought or dry weather adapted or microbes. So these will be selected for. There is some evidence that these drought tolerant microbes are more stable. The community won't change. Once it hits that drought community, it'll be pretty stable. Okay. So there have been quite a few studies of the effect of drought on the microbial community compared to too much precipitation. There's not a lot of studies on that. And what they find, this was a study looking at 8 different sites across grasslands where they had drought treatments. So they had ambient water going in versus a deficit of water going into the soil. And then they measured the microbes in both of these treatments. And what they found was there really wasn't a huge difference in bacteria or saprotrophic fungi. That's what this SF is. But arbuscular mycorrhizal fungi were much more abundant in terms of biomass in the dry treatments, which could hint to an important role that they play. There have been studies looking at when plants will kick out these symbiotic fungi and bacteria, and it's when they're not needed. So there have been quite a few studies looking at additions of fertilizer to soil. And what that causes is kind of the plants to kick out their microbial associates, because if they're using them to get nutrients from the soil environment, if the nutrients are suddenly readily available, then they won't need their symbiotic organisms. And the same thing could be said here. Could be argued that the arbuscular mycorrhizal fungi are increasing their drought resistance, maybe giving them better access to water or better photosynthesis efficiency. Less water is lost. It's not really, they didn't go into that detail. Okay, so here again, we're looking at arbuscular mycorrhizal fungi. The EMF refers to the ecosystem multifunctionality. And so this is a metric that it's kind of subjective. The author, the researcher, creates it based on whatever metrics they measure. So in this study, they were looking at effect of drought on the arbuscular mycorrhizal fungi and the response of the plant. So For this study, to create this metric, they have the plant community biomass as well as diversity, and then some nitrogen. So nitrogen use efficiency, emissions of nitrogen to the environment by the plant. So they measure these metrics in a non-drought treatment, which is this ND versus a drought treatment. And then they also had mycorrhizal treatments here. So M means it got arbuscular mycorrhizal fungi. NM means it did not get arbuscular mycorrhizal fungi. And then you have non-drought and drought. Okay, so from this, just pay attention to the red and the blue. You have an increase in ecosystem multifunctionality, so you have less of an impact when you have mycorrhizal fungi. And then the same was found here, where we have, instead of pre-drought, during the drought, and then recovery here. And the recovery during the drought was not as harsh as when you didn't have mycorrhizal fungi. Okay, so I was unsure how familiar everyone was to ordinations. I was wondering, is everyone here familiar with ordinations and how to read them?
I'm definitely not.
Okay, good. So an ordination is something that's used in microbial ecology to take this multidimensional data set that we have and make it easier to look at patterns. So what I mean by this is if we are sampling in a community, so here's a location in South Texas where I sampled 3 samples from below these mesquite trees and 3 samples from the grassland. And so when I take a sample, I extract the microbial community that was within that sample. So you end up with a data set that looks like this, where each row is a sample. So I have my 6 samples here, the 3 mott's, which are the blue ones, and the 3 grassland samples, which are these red ones. In each column is the species, and within each column, we have the abundance of that species within that sample. So we end up with this data set that can be huge. For bacteria, you can have thousands of species. For fungi, you can have hundreds. And it makes it really hard to discern patterns. So we use this tool called an ordination, where each point represents a single sample, and we use a metric called a dissimilarity metric. So we'll compare all of the samples here. How many species do they have in common? So all of the samples except for this grassland 1 has the species 1. And then so how many species it has and how abundant they are goes into this metric. And so when you look at these, all you want to look at is whether the points are closer together, which means they're more similar. The communities have more species in common and they're more similar in abundance, versus if they're further apart here. That means they're more dissimilar. So they have less species in common and they're less abundant. Okay. So I'm going to be showing a couple of these ordinations. If anyone has any questions about them, you can unmute and feel free to ask me. This is where I think a lot of people have a hard time understanding. Okay, so this was a study I did in grad school where I sampled ectomycorrhizal fungi, so on the roots, and then endophytes in the leaves of ponderosa pine. And this was on one mountain range that I sampled in Arizona, where we sampled from a low elevation to a high elevation. And this was to look at changes in climate as you go up, kind of removing the effect of distance, which can have a really strong effect on the microbial community. So first, looking at the ectomycorrhizal fungi, what we found was we saw distinct communities that shifted as you moved upward in elevation, so going this way. So these were the low elevation communities where it was drier and warmer, and these were the colder weather communities. Compared to endophytic fungi, though, there was really no structure. There was no difference in the communities. The same species at the same abundance were at high elevation and low elevation. And so the reason I wanted to show this was just to highlight when you're thinking of the microbial community, which there are different communities, you can see different responses to climate change. So the microbial community won't always respond the same way, which makes it a little bit difficult. Okay. So now I'm going to shift to the plant community and talk about the role they play with microbial communities. So as I've mentioned, I did a lot of my research in South Texas, and these were mesquite savannas. So I'm going to be talking about those ecosystems. I haven't done a lot of work in blackland prairies, but some of it will be applicable to the blackland prairies. Okay, so in this ecosystem, you have these distinct woody patches or mottes filled that are kind of started by a mesquite plant, and then you have other plants come up around it. It acts as a nurse plant. And then in between them, you'll have grassland. And so you have this really close set of plant communities that can be either really far apart or really close together, and it's heterogeneous across the landscape. So when we went in and sampled, we found a really strong difference in the fungal communities in both the woody patches and grassland, even though they were really close together and it was just throughout. There was no similarities hardly. And we found the same thing within the bacterial communities. So you can have really large changes in a really small space within the soil community. And these were taken from bulk soil. So this is bulk soil communities. And these correlated also to differences in function. So here we have 3 different enzymes. First one dealing with carbon cycling, second one with nitrogen cycling, and the third one with phosphorus cycling. And then you'll have 2 box and whiskers within each, one for grassland and one for woody patch. And you'll see higher enzyme function within the grasslands for each of these compared to the woody patches. And so what's driving this? If you remember, mesquite is a legume, so it's nitrogen-fixing bacteria on its roots, increases nitrogen within the woody patches. And we found that was true for most of the nutrients that we looked at. They were much higher in the woody patches than in the grasslands. So this higher function could be within the grasslands, could be the microbial community making up for this deficit of nutrients that are available. So you can see that even within close distances, you have different communities that are functionally very different as well. So these were the 6 nutrients that we looked at. And then when we looked specifically at arbuscular mycorrhizal fungi, we found a similar pattern in which they were much more abundant in terms of species richness in the grasslands and then overall abundance in the grasslands. And you had slightly different relative abundance of the different communities, different genera that were present, excuse me. So you can see a really strong interaction between both plant community, nutrients, and the microbial community. Although here I was not able to determine cause and effect because it was an observational experiment. But in South Texas, they're having a really hard time with guinea grass. I'm sure some of you are familiar with it. It's a pretty bad invasive, produces a lot of biomass. It can change a community really quickly. So these 2 pictures actually aren't taken too far away from each other, but you can see the communities are very different. So how does invasive species such as guinea grass impact the microbial community? So here I'm showing you some nutrient differences between invaded and uninvaded. And what you're seeing is a slight increase in the invaded grasslands. So here I'm only showing data from the grassland. I think I should specify. And so this is pretty common phenomenon that happens after a disturbance, and invasion can be seen as a disturbance, but also like if you go in with a plow or if you do like a prescribed burn, you'll have a quick influx of nutrients in the soil that are easily available to plants, which is why I think people do them, because you see this really great response right after. And then we did see a huge difference in the fungal communities in invaded and uninvaded grasslands. So the question then became, when you have this change in the fungal community, and this was also mirrored in the bacterial community as well, but I'm biased toward fungi, so I'm only showing fungi here. The question became, when you have this change in the microbial community that's so obvious in the composition, how does that impact the plant community? Does it have an effect or does it not have an effect? And so what kind of drove us to look at this is we were noticing that in areas where guinea grass had invaded, you would have these bare spots where it didn't seem like competition was keeping out any natives from growing. But you still weren't seeing any native seedlings come up at all in these bare spots. So that gave us some thoughts like maybe there's some allelopathy, maybe guinea grass is producing a chemical. And then me being interested in fungi, I was interested in, was this something to do with a plant soil feedback? So plant soil feedback is a phenomenon that's been pretty well studied now, but it's where you have this feedback mechanism where the plant is producing litter that will go into the soil, is producing root exudates that are moving from the roots into the soil. And in doing so, it's kind of conditioning the soil environment to be more beneficial towards it, or less beneficial. So this could be through selection of particular microbes, or this could be through releasing a chemical, like a secondary metabolite. So we did a quick experiment, and we pulled soil from invaded and uninvaded grasslands and planted guinea grass. And the results were very obvious. In invaded soil where we planted guinea grass, it grew substantially taller and had more biomass versus soil from uninvaded grasslands where it was much smaller. And so in invaded soil, guinea grass was both taller, had longer roots, and overall biomass. And then because we had collected soil, we had a pretty good collection of the seed bank within soil. So we let some pots go and calculated what native plants grew out of the soil from invaded grasslands and soil from uninvaded grasslands, which I'm going to show here. We grouped them broadly by functional groups, so dicot versus monocot. A lot of them weren't fruiting, so we weren't able to get them down to their exact species. But we did find an uneven effect. So there was no difference in what dicots or forbs came up in the uninvaded and invaded soil. But when we looked at monocots, we saw a much lower growth of grasses from soil from invaded grasslands versus uninvaded grasslands. So there did seem to be a positive effect on itself in invaded grasslands where guinea grass was selecting for itself and kind of preventing other grasses, which would be its main competitor, from growing in areas where it had previously invaded. Okay. So shifting more now into disturbances, looking at frequency and intensity. Okay, so intensity and frequency of disturbance does factor into the effect that it will have. More intense and even low intensity disturbances that are more frequent will have a stronger effect on the microbial community and plant community for that matter. Some disturbances can be good, like I mentioned. They can cause like this quick influx of nutrients that are bioavailable to plants. So this is where prescribed burns, which have been used for hundreds of years, really are beneficial. They keep a lot of the invasives in check, and they can kind of renew your microbial community. If there's some competition where one is becoming more dominant, it can just provide a quick reset, which can sometimes be good. And then microbial communities do go through succession. So if you have an old community and would like to reset it, again, doing a quick disturbance sometimes helps. Okay, so this was another study I did in Arizona. So I had two mountain ranges that were at similar latitudes. The Santa Catalina Mountains, which are just north of Tucson, and the Pinaleno Mountains. And so both of these mountains had experienced a pretty recent burn at the same elevation, and it was kind of nice how parallel it was. But I wanted to see when you have similar environments, so similar climate, similar plant community at that elevation, and similar disturbance, do you see similar recovery in both of these communities? And here I was looking at ectomycorrhizal fungi. These are some of the root tips that I pulled out and took a photo of. And so what we found was in burn sites, we had higher diversity, which is also something that can happen where you have an increase in diversity, because you'll have these fire adapted species coming in, and then you'll still have, if it wasn't a super intense fire, you'll still have some of the community from pre-fire in there as well. So you can have an increase in diversity, which is good. But what we found, this is another ordination, was that the communities in the burned, which is the black, and the unburned, and the two mountains were completely different. So in the Pinaleno Mountains, the burned community was just a smaller subset of the unburned community. And this can just be that certain species were more abundant than others, but pretty much similar species were found in both. In the Santa Catalina Mountains, you have a complete shift in the burned and unburned sites in terms of species composition. So in researching the Blacklands and thinking about them, it's been highly modified and manipulated. I'm sure you guys are all aware, but you had controlled burns done by the First Nations people who were here. For how long, no one knows. And then after that, you had heavy farming and grazing by immigrants to the area. And then now we have this huge population growth, which I think we heard about last time. So my question with this when thinking about restoration is, is there truly a native microbial community that can be referenced for restoration? Or is it kind of unknown? And that I'm not sure about. I'd be interested to hear your thoughts in the question section about whether there is a true native Blackland prairie that could be referenced. But in looking at restoration, some of the most common methods that I'm sure many of you use are herbicide applications for invasive species removal or mechanical removal, soil preparation, so tilling or burning, and then sowing native seed. But a lot of this doesn't really take into account the microbes, and that's understandable. They're a cryptic community that is very complicated. What's becoming really popular in the literature is this single soil inoculation. And so they choose a donor site. And so this would ideally be something that's local. And then also something that is the community you are striving to have within your donor community or your control site, your site that you are inoculating. So this is from a study where they had a control site that they inoculated with 2 different inoculums from 2 different ecosystems. So we had a grassland inoculum and a heathland inoculum, and then they measured it to see where it ended up. So these are the ideal ones that they're trying to aim for. So they're trying to shift this control microbiome to this donor grassland and donor heathland community that they sampled from. And so a lot of times though, this is what you see is not a complete shift. It moves in the direction. So here we have vegetation, here we have microbes, and they are shifting away from the control, but it's not exactly the same as the donor community for both the vegetation and microbes. And I don't remember how long this study went on, so it could just be like you need more time to have it reach this donor grassland ideal. But this is something that's been done more and more often. So some considerations are if you do decide to do this inoculum, that you need to do it in parallel with sowing native seeds. As I hope I've shown, the plant community is really important in selecting for the correct microbial community. And then these deep-rooted prairie plants that you naturally find in these blackland systems are great at improving soil structure, so they will just do their work. Additions of organic matter would be helpful for the microbial community. So last month, Brian mentioned, if I can remember, in the question, someone asked specifically about restoration, and he said he had heard somebody had planted a cover crop and then mowed it down the next year and then leaving the roots in place, seeded it on top of that. And that would be great in terms of organic matter addition and keeping the soil structure intact. I couldn't find any references to that specific methodology in the literature, but if anyone has heard of someone doing that or knows of a paper where it was done, I'd be really interested, because I would imagine that would be really good for the microbial community. And then also choosing a nearby native prairie as a source of natural inoculum is key. So some issues with this method of using soil inoculum is you are essentially going to disturb a native area. So that could, cause some issues later on. It could cause non-natives to gain a foothold or just disturb the microbial community enough to where it's not really a good inoculum anymore. There has not been enough study on that particular aspect of this method yet. So I can't speak to that, but it is something to be cautious of. I've been very curious about that. Additionally, the complexity of microbial communities, they're very complex. I hope that this has convinced you that anybody trying to sell a single microbe as a solution is a snake oil salesman. You can't fix this problem with a single microbe. It has to be a community. So that's why the soil inoculum as a method is probably better than just a single inoculum. And then mismatch with local conditions. So wherever you pull from, you want to make sure that you have similar conditions as what you're going to find locally. Or it can lead to a mismatch where you don't have the best symbiotic microbes that can go with the plants that you're planting. Another issue is just the scale and cost of moving and distributing soil. I'm sure you guys probably have experience with moving soil, but it gets really heavy. When I was sampling soil for some of these studies, it was a workout. And then there's uncertain long-term outcomes. Many of these studies that have been done on the soil inoculation method as a restoration method aren't super long. There's one study by Wubbs, WUBS, who I would argue is the authority on this, that I think was 14 years long. And there were good results, but it wasn't the same as the reference community or the donor community. And then you can still have inconsistent results. This is a living community. It will change. What you select for may not be what you really want to select for. So just keep that in mind. Okay. And then lastly, I just wanted to thank University of Texas and the Brackenridge Field Laboratory. They're working on a number of field stations for UT. Really good work. And then Lee and Ramona Bass, who funded a lot of the research I've done in Texas.
Thank you, Dr. Bowman. That was really interesting. I think we do have some questions, and I think probably one of them that has already come up is soil inoculation. We've heard it, I've heard of it in terms of just like in a garden, if you're trying to get a particular plant to grow, like say if I go out into my landscape and try to transplant something from the landscape into my garden, right, to cultivate it there. It would be good to bring some of the soil along.
Yeah.
There. So in a small scale, but what more do you know about any kind of a larger scale inoculation? And yeah, I think the idea of what do you go dig up something else? I mean, that seems
Yeah.
ethically challenging. What do you think about that?
Yeah, I've had some issues with it. One of these papers by Dr. Wubbs, who I was referencing, it was the 14-year study that they just took like the very, very top soil. So like not even a lot of soil, and use that as like the inoculum. So ideally you don't need that much based on this one study that he has. But in that study, I don't think that they took samples of the microbial community, so they couldn't track those changes. But yeah, it is something I'm super curious about, because if you're going to do, a whole, even just an acre, that would be so much soil. I don't think ideally it's a very tractable method for moving the soil, for distributing the soil. When you sample it, you're going to be disturbing it. That's a disturbance. And so that can change the community a little bit. But yeah, the whole question of like what happens to the donor community, is it just sacrificed? I don't know. I don't think there have been any studies that have looked at that yet.
So interesting. All right. Well, Marvelyn, take a look out there, and everybody, feel free to put something in the chat or just unmute and ask Dr. Bowman a question.
I see one question
here.
about how soil inoculation is done.
Mhm.
So, I would imagine that you could either mix it with your seed and broadcast it out that way. You could also just have, I think those little fertilizer carts, I forget what they're called, and put it in there and it would broadcast it as well, would be the best method. I think that would be the easiest method than going in and like tilling it in.
Well, I think often the advice is that you don't want to over disturb like more than the top quarter inch of a restoration soil. So I think to your point of it feels like one is being sacrificed for the other. And is that going to really be useful on the new land, because now it is disturbed? So you're bringing in disturbed soil. So
Yeah.
I'm not sure that even if it was healthy on the first place, is it going to still be healthy in the second place? So that will be interesting.
One idea that we've kind of toyed around with, because we're looking to do a restoration experiment in South Texas where the guinea grass is, and we wanted to incorporate a soil inoculation treatment, is just making our own donor community. So that would be just like a field that we would plant whatever plants we end up wanting to plant in the restored field, and kind of condition the soil for a number of years. We don't know how long that would take, or if it would even work, but that was the only method we could come up with where you wouldn't be disturbing like one of the few native blackland prairies or whatever.
Right. Yeah,
To
sort of a massive compost pile.
yeah.
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 So somebody had a question too of the impact of herbicide on a microbial community. So I know that that's been discussed a lot. And do you have a stance on that?
Yeah, I mean, it's hard not to use it sometimes, especially with some invasives. Some of them are impossible to get rid of without herbicide, honestly. But it will have an effect. It kind of depends on like what kind of herbicide you're using. But it likely will have a small effect. But microbes, especially bacteria, are really good at degrading chemicals. And especially bacteria, because they're unicellular and reproduce really quickly, they can evolve to break down a lot of chemicals. So maybe you'll select for microbes that can break down those herbicides, but it's kind of theoretical.
And it may depend too on your microbial health before you spray, how fast that can process through.
Yes, definitely. Like some of them are designed to break down really quickly in the environment and not stay around, which are probably would be the best ones to use.
So while other people post their questions, I had one about uh, is there a max out of the microbial benefit after a prescribed burn, similar to what you described, or how it sort of reaches a maximum with a lot of water? It likes the water, but then at some point it's like, okay, I've had enough water. Do you see a similar response after a prescribed burn or a number of burns?
Yeah, so there's actually been quite a bit of research on the effect of burning on the microbial community, and that's just because it's used so much, not only in restoration, but for like logging. Um, a lot of logging operations will burn to clear the land, but they want mycorrhizal fungi there because they help the trees grow faster, help the seedlings establish. So there was a lot of research into that. And so there is like prescribed fires because you do them when you don't have a huge fuel load. They're usually lower temperature, and then they move very quickly over the ground. So they aren't staying in one spot. So prescribed burns shouldn't have a huge effect, but you wouldn't want to do them every year. I'm not sure. It probably would depend a lot on the ecosystem of how frequently you would want to do it. I know that the Lady Bird Johnson Wildflower Center, they have a big study on prescribed burns. I bet they have a lot of data on the microbial community and how it's affected by frequent burns.
They just did one last year, so yeah, that would be
Yeah,
a good place
so
to check.
yeah, I think if not, then they should, because they have a lot of really cool data there.
Right, right. So someone also asked if you have a resource list for some of these studies that you've discussed this evening.
Yeah, I can send one if that's okay to you, Connie, and then you could disseminate to everyone. Okay, yeah, I can do that.
Sure. And, you know, like Dr. Wubb's research, is that like maybe available on Google Scholar? You could search for it that way, or
Yeah, you
do you
should be
know?
able to. If any of them are that you're interested in or unavailable, if you just shoot me an email, I can download it and send it to you
Oh,
guys.
that's so cool. Yeah. It's good to have a resource for that. When,
Yeah,
when you hit that wall, you can't get it. Yeah.
I can tell
Okay.
you how
So
frustrating
yeah,
it is. Yeah.
I know. So yeah, if you guys out there, if you haven't looked at Google Scholar, just put in Google Scholar. It's an indexed repository of academic papers and research on all kinds of things. So you can search on any number of topics. You can find some really interesting stuff out there.
So I'm seeing one question about the challenges of invasives and the changes to the microbial community in the face of climate change. And yeah, I would say invasives are a very interesting challenge in that, you know, I feel like each invasive plant has its own method of invading. Some use, secondary metabolites. It just varies by region and where they're from. So the methods of kind of overcoming this invasive can vary by which invasive you're talking about. So the main ones in South Texas are guinea grass and buffalo grass that we're dealing with right now. There are, of course, many others, but those are the two main ones. And guinea grass produces this, kind of negative plant soil feedback for its competitors and positive for itself. But buffalo grass, we haven't really found any evidence of that type of interaction. And so we aren't sure yet how it's becoming so invasive versus guinea grass, we kind of have an answer for. And so it comes down to like, how is an invasive invasive? And so, yeah, it can be very challenging, but I think it's only a challenge to the microbial community if it comes in and becomes like this monoculture and you lose that variation in the soil structure, like what litter is being put in. It's all from the same plant when there's an invasive that takes over. The root exudates are all the same across a landscape, where before, if you had a really healthy, diverse community, you get this like heterogeneous distribution of root exudates and plant litter that can create this really diverse niche space for microbes. But when you have like these monocultures of invasives that come in, it changes everything. And then with buffalo grass, it changes the fire regime, which can further damage the microbial community. So yeah, there's a lot of challenges.
It seems like in the future there could be something about, you could vaccinate your land with the soil of your land in such a way that it would not allow the invasion. We would work not at the leaf level and top killing and root killing, but we would work at the soil level so that invasives just couldn't take hold there. That would be so cool. Yeah.
Yeah, that would be really helpful. I think part of the issue is like we study a lot of the grasses that come from Africa, and when they get here, they just don't have, there's no herbivores that eat them. There's no pathogens. Like in South Texas, there would be just these whole fields of guinea grass, just so abundant. You would think there has to be a pathogen on this somewhere. There would be no pathogens. It would be spectacular.
Mm-hmm.
So they don't really have any enemies, which causes them to increase. And then there's this one theory I'm working on a paper on where you, because you also lose pathogens, but you also lose your beneficial organisms, so your mycorrhizal fungi. And so you don't have that carbon cost either. So you have no carbon that you're putting towards beneficial or detrimental microbes. So you can invest it all in growth.
Mm-hmm.
Someone asked about the plastic research.
Yeah.
Yeah, I can talk a bit about that. So this was my foray into industry. I've been in academia previously forever, and so it was a huge culture shock. But the company, we are starting off trying to degrade diapers with fungi. And so diapers can be recycled. They have about 5 different types of plastic in them. And because, they have this mix of plastic and they contain feces, human feces and urine, they can't be recycled. So they are this waste product that everyone uses because they're extremely convenient. And reusable diapers are horrible. I've used them before. So this was our idea to kind of combat initially the plastic crisis with diapers. They stay in landfills for over 400 years. I mean, really, we don't know how long they'll last because we haven't had them around for that long, but they do not degrade easily. So that's what we're starting with, and then hopefully we'll expand into other plastics, non-recyclable, single-use plastics as well.
Wow.
Yeah.
Well, okay. Well, Dr. Bowman, thank you so much again for your research. You are saving the world, and we we really appreciate you taking some time to help us understand better just the importance of that community in the soils and the complexity of it.
Awesome. Yeah, I'm happy to answer any questions, and I will send a reference list probably this weekend sometime.
Okay, that's perfect. We appreciate that.
Of course. Bye, everyone.
Thank you so much. Thank you, everybody. Good night.